most people think of honey, they think of a sweet natural food. But in historical accounts and older scientific writings, honey has sometimes been described as something more, a substance capable of preserving biological materials. In this video, we revisit one of the most intriguing texts on that subject, Dr. Stoimir Mladenov's article on the preservative effect of honey. The central question is simple, but scientifically important: if honey really preserves, is that effect caused only by its high sugar content, or is something more complex happening? Long before Mladenov, honey had already been linked to preservation in historical memory. References from ancient Egypt, Assyria, and Greece helped shape the idea that honey could slow spoilage or protect biological matter. What makes Mladenov interesting is that he did not simply repeat those stories. He tried to turn an old belief into a scientific question. In other words, he asked whether a traditional observation could be examined experimentally and explained biologically. According to the article, Mladenov used several natural floral honeys collected from different regions of Bulgaria. He placed seeds and a variety of biological materials into honey, including kidney, liver, eggs, and other samples. Importantly, he also created an artificial honey control made mainly from glucose and fructose. That comparison mattered, because it allowed him to ask whether preservation could be explained by sugar concentration alone, or whether natural honey contained additional active factors. Mladenov reported that seeds stored in floral honey for 1 year retained normal appearance and strong germination. He also claimed that several biological materials, including kidney, liver, and eggs, remained preserved much longer than expected when kept in natural honey. By contrast, the artificial honey control performed worse. These are the most striking parts of the article, because they suggest that natural honey behaved differently from a simple sugar mixture. The real significance of the paper may lie in these comparisons. If sugar alone explained the preservative effect, then artificial honey should have shown similar results. But Mladenov reported that it did not. He also wrote that when honey was heated to boiling, or above roughly 80 degrees Celsius, it lost its preservative effect. At the same time, when honey was neutralized to around pH 7, it reportedly retained some of that activity. Together, these observations pushed him toward an explanation beyond sugar alone. Based on those results, Mladenov rejected the idea that honey was simply a concentrated sugar syrup. He proposed that biologically active plant-derived substances were involved and referred to them as phytoncides. In the scientific language of his time, this was a serious attempt to identify a living, active principle inside honey. Today, the term may sound dated, but the key insight was important: honey might preserve because of a combination of active properties, not just because it is sweet and concentrated. Modern science would describe the situation in a more detailed and multifactorial way. Today, honey's antimicrobial and preservative effects are generally linked to low water activity, high osmolarity, natural acidity, hydrogen peroxide generation in many honeys, and a complex mixture of phenolic compounds and other bioactive substances. In that sense, Mladenov's intuition was partly ahead of its time. He was right to suspect that sugar alone was not enough. But modern research explains that effect as a combined physicochemical and biochemical system, not as one single active substance. Even by today's standards, the paper has real strengths. First, it used an artificial honey control. Second, it tested a wide range of materials, from seeds to animal tissues. Third, it did not focus only on appearance, but also discussed germination and bacteriology. And fourth, it tried to explore mechanism by asking what changed after heating or neutralization. For an apitherapy-oriented paper from 1967, this was an ambitious and thoughtful design. At the same time, the article has important limitations. In the version available today, there are no clear sample sizes, no detailed sterile methods, no full replication data, no statistical analysis, and no detailed chemical characterization of the honeys used. Some of the outcomes are described in observational terms rather than with rigorous quantitative measurements. So this paper should not be treated as final modern proof. Its value is historical and suggestive. It opens a research path rather than closing the question. The importance of this paper lies in the bridge it creates. Mladenov stood between historical observation and modern biomedical thinking. He took an old idea about honey seriously, tested it with controls, and tried to explain it mechanistically. That makes the article more than a curiosity. It is an early and meaningful contribution to the scientific imagination of apitherapy, tissue preservation, and honey biology. This presentation is based on a critical revisit of Mladenov's original article together with later scientific literature on honey's antimicrobial and preservative properties. The historical source remains central, but it is read here with modern caution. Where a statement is presented as something Mladenov reported, it should be understood as a historical claim rather than definitive modern evidence. The most valuable next step is not simply repeating these observations, but testing them again with contemporary microbiology, histology, and biochemical methods.